good. Make sure the red one is on. Okay, we're gonna have a class today, so not gonna be too long. We will be to do some of the testing of the biochemistry test 2 and do some new stuff. Okay. So first of all, and everybody should look at your SIM agar. Let's go over something. SIM agar, which is for sulphide. I is for Indo, and M is for motility. Okay, everybody look at your slants, SIM slant. I wanna get one sample. Who has Proteus vulgaris? Can I see the Proteus vulgaris slants, what it looks like? Okay. Who has E. coli? Or who has Pseudomonas? Does anybody have Pseudomonas? Can I have a Pseudomonas slants? Yeah. Thank you very much. Now, if you compare here these two, what you can see? We could see that it did not really turn to black, but there's a little bit of black showed here for Proteus vulgaris. This is Pseudomonas aeruginosa. Most likely it's not black. It may be the temperature is the reason, because we put in the room temperature. It might be the other reason. So you can see this Pseudomonas aeruginosa, it's not black, and this one is slightly black. So we will say from here the sulphide, the Proteus vulgaris is positive, and the Pseudomonas aeruginosa is negative. Because this should be turned black, and here we see it is like a weak black. Okay, ideally, and you can look at your own. If you have an E. coli, you can decide it by yourself. Now, the second thing when you look at these two differences is talking about motility. Okay, I want to talk about motility first. Here you can see this is Pseudomonas right in the middle. And the Proteus vulgaris, you can see it goes pretty much more area compared to the Pseudomonas. Okay, so we will say Proteus vulgaris have a strong motility, which is better than Pseudomonas aeruginosa. Okay, now ideally this should be turned very black, because Pseudomonas Proteus vulgaris, this is the Petrotrichus. And this is the Petrotrichus, which means the flagella is covered the whole surface, and the Pseudomonas aeruginosa is monotrichus. Only you have one tail on one side. So that's why Pseudomonas aeruginosa is have a very limited motility ability. So which means it's going to be stay in the middle, just like in the line in the middle. Okay. I'll return this one to you, and return this one to you. Now my next question is how we going to test the Indo. Okay. When we tested the Indo, I said we need to add a reagent. This reagent add on the top is called a Kovac. Who has E. coli? Can I have one slant of E. coli? Yeah, same the SIM agar. Okay, can you do this for them? I have a Kovac right here. Can you add a couple of drops on the top? I just want to do a test for E. coli. Okay, we'll see. Couple drop on the top. Who else has E. coli? Yeah, just grab the E. coli. And add a couple on the top. That's good. Okay. Now wait for a little bit time. Then let's have another sample. You guys have Proteus vulgarius. Let's try the Indo too. I want to test this one whether it is really positive or not. Because sometimes it maybe get contaminated. Okay. It might be get contaminated. See that Proteus vulgarius right here. Who has this? You had a Pseudomonas aeruginosa. So let's do that too. So we can see whether it's good contaminated. Okay. This one seems like it's okay. Now obviously you can see there is some contamination for the Proteus vulgarius, and you can see that tube. Is that a little bit pink?
Yeah.
Yeah. It should not be pink. This one did not show that pink, but E. coli. Can I have one E. coli? I have one. I have one. Okay. This one give it to you. Okay. Now you can see a very obvious color for these two E. coli. This is showing you cherry red on the top after you add Kovac. For this one, for Pseudomonas aeruginosa, there is no color there. And we also tested, I suppose that is Proteus vulgarius, but what you see there, it's actually not that colorless. It's still cherry red. So I think that one might be getting contaminated to sample. Okay, so here, I just tell you the results. For Indo test, it should be E. coli. Is absolutely is positive, which is cherry red color. And I tell you one thing, this is a very quick test for E. coli. Is use Indo test. When you use SIM, and then you add Kovac reagent, and you see it's a cherry red color. Because for E. coli, there's a lot of tests. We didn't do in the lab, but for example, like 3-tube fermentation test. Like next week we will use petrofilm to see the colony. We use spread plating, whatever. It takes a very lot of time, including the entratube. Is that right? But as quickly as this, in some of the clinical lab, use Indo test, we can identify E. coli very quick. Okay, so E. coli adding Kovac is Indo test positive, is pink to cherry red color. Okay, so that's what we finished right here. You can do it by yourself. I'll just give you 2 examples. Okay, this is the first one. The second one is a phenylalanine deaminase test. So we are doing the confirmation. This deaminase test, what we did is that we want to see where is the phenylpyruvic acid generated. So we did the zigzag streak. We're gonna add is FeCl2, ferric chloride. Okay. Now I have a ferric chloride here. Let's do some tests. Who has Proteus vulgarius? Okay. I'll add for you. We add some, a lot actually. Then you shake it a little bit. Shake it a little bit, okay. Who has let's do Pseudomonas aeruginosa. So can I have 1 ounce? Yeah, that's good. I'll be first. We'll see what it is. Then you shake a little bit. Shake a little bit. And can you let them do an E. coli? Okay. Now let's get those samples to see is there any difference. Then you have it. Let's have that one. Okay. Here you can see some of the difference between that. Can you see for Proteus vulgarius is pretty dark green. For Pseudomonas aeruginosa is actually original color, a light yellow color. So which one is positive? Proteus vulgarius is phenylalanine deaminase test positive, and the Pseudomonas is negative. Okay, so pretty obvious you can see the true difference. I actually would love to keep these two. Is that okay? And I want to be doing some picture for the student who need not come into here. Okay, so I'll keep these two here. So here you can see Proteus vulgarius is positive because it's dark green, and Pseudomonas aeruginosa is negative. Okay, so this is second test. Okay, third test I want everybody to do is called Mrvp test. So you should have a tube. I want everybody to do that. Is Mrvp confirmation test. Okay, how to do that? In this group, you guys should have one or two. You should have one solution. Is that right? One tube. We inoculate, which is either E. coli and Enterobacter. So let's say you have a solution which is EC. What we're going to do? You gotta grab an empty 5 ml tube. Okay, you give them each of them have one. Who has this? Who going to do it? You're gonna have one. Okay. Then, yeah. So I'll be. You don't have a solution. Okay, I didn't see so. Yeah, give them each of them is an empty tube. That's a 15 ml tube, but we. Sorry, I forgot to give you go. So, what we're gonna do? You're not gonna see anything there, okay? Because it's just a growing. What I want you to do is that. Let's say this is an E. coli solution. You just add a little bit. We don't use like pipettes or something. You just dump a little bit, about 2 to 3 ml into an empty tube. And you mark the original tube as MR and the new tube as VP. Okay, you do that job first. Let's do that right now. Just dump it about 2 to 3 ml into the empty tube, and the mark the original one is MR and the new one is VP. So ideally we split them onto 2. Okay? Now I want you to do that for them. Is everybody, okay, you did that, is that right? You guys all did it. Now what we're going to do, for Mrvp, we have two separate tests. This is MR is to testing end products which is acid, and that is a end product which is neutral. So we're going to add a reagent. So first of all we add MR reagent. So you go ahead and add them a little bit. For them, the MR tube. Whatever you have the tube, which label is as MR, Rachel gonna add the MR solution for you. So you add for them a little bit. You can borrow from somebody. You can open it? no, you have to dump a little bit on the tube. Dump a little bit on the empty tube. You can get bottles out. Yeah, and more add a little bit drop. Okay, see? Okay, then you close it. Close the cap. Yeah, Rachel add them for whatever the tube is, set MR. Okay, so MR test. We have E. coli and the Enterobacter. Okay. Who has E. coli? Can I have E. coli? Just an example. Okay. So when you add it, always remember when you add it, you have to shake a little bit. Okay. Not on the top. You have to shake a little bit. That's very important. Who has Enterobacter? Okay. Who has Enterobacter right here? You guys all have E. coli or Enterobacter? You have the Enterobacter. Okay. Give it to me. Okay. Can you see the difference? This is E. coli and this is Enterobacter. What's the difference? E. coli is pink and the Enterobacter is colorless. So what that means, E. coli is MR test positive, Enterobacter is negative. Okay, so I'm going to return these two to them. And you return this one back to me, and I'll return this to them. Okay, so this is the MR test, Mrvp test part. Just write it in Mrvp. Okay. Okay. We're going to do VP test. VP test is gonna be a little bit wild. It may not show any results. Maybe showing. So VP test. We want vocus per scale. We want to test it whether it has the end product which is neutral. Because usually it's generated acid product first, then become neutral. So it's gonna be a while. And we will do add VP reagent A and VP reagent B. So Rachel, you go ahead to add for them. Reagent B, I'll add reagent A. Okay? Now, let me have you tube, whichever tube you label as VP. Okay? Now you have to shake a little. Shake a little. You may not see anything right now. So Rachel will add reagent B. Then you shake it. You may not see anything right now. What I want you to do, if you didn't see anything right now, put on the rack. And then we wait at the end of the class, or we maybe wait next week. Okay? It takes a while to become pink. It should become a pink. So I tell you when we're saying for the VP, it should be opposite. So the VP results should be E. coli is negative and Enterobacter is positive. So which means this one should be pink, this above one is not pink. Remember IMVC test? So in the Mrvp, vocus per scale and citrate, and I wrote that positive, negative, positive. So that's a way to differentiate that. Okay. It's pretty simple, not that difficult, but I just want you to learn about something. Now, this one I will be very briefly. I'm not gonna go very detailed. Okay. What you should have. Everybody compare with your bench partner. You should see. It should be the color is the same. If you and your bench partner compare, every color is different, which means you get some contamination because we all inoculate is E. coli. So first of all, you compare with your bench partner. Is they all the same? Okay, you can have one. That's what I did. No worry, you can have one. Okay, so first of all, compare. Is there any difference? If no difference, that means you guys did good. If any difference, which means you should have a red, then you have a yellow, that means you get a contamination. So that's the way to test your contamination. Okay, very, very simple. Okay, so just compare, okay? The second thing, I'm going to be a little bit easy, not going to be too complicated. What we have to do is that each of them, originally, you see so many wells, is that right? Okay, I just give you 3 examples. For example, let's say we have a glucose. We have a lysine, for example. Then we have a ornithine. Okay, then we have a sulphide and indole. Now, first of all, you need to know, these type of indole tests, you have to do it separately. There is no way you do that, because we have to add a Kovac, is that right? That you have to do separately. So let's say here is positive. How do you know it's positive or negative? You have to compare standard color sheet. So we're gonna just skip that. I'm not gonna give you color sheet that you to compare. It takes too long time. We have limited time. I just usually if it's normal class, I will everybody give a sheet that you compare. We just skip here, okay? I just let you know how we do it. We compare standard color sheet. If it's positive, we record it positive. Let's say positive, then positive. Negative, negative. For example, then we record it. Then each positive or negative, there is a code. This one may be code number 1. This one may be code 2. This one may be 2, maybe 3. Okay? Then all these codes, you're going to have to add up. So this maybe add up 7. Then here you maybe add up 5. Then another 3, you maybe add up 6. This will be your standard code. And this standard code, you have to compare a code book. If it's matched, which means the species, the genus will be matched. Okay, so use these to compare a standard code. I just tell you how to do it. We don't have to do it. Okay, then if the code is matched, even one digital difference, there might be miss a genus name. So if it's matched, which means that's what the genus name is. But it's only go to genus name. So let's say salmonella, there's so many salmonella, it's only go salmonella SPP. Okay, so that's what the entry tube do. So I just give you a demo, we're not gonna do it. It's take too long time. Now I said this is gonna be a gift, so you can take it home. If you like. Okay? If you like, you can take it home. As long as you didn't open it, it will not get contamination. It's very beautiful looks, the different color. Okay. So that's what we have. We do some of the review from last week. Okay, we're gonna go to today's lab. Okay, today's lab is pretty straightforward, but we're gonna talk about some clinical lab. If it's a normal time, I usually have another clinical lab. I just let you know how we did it before. So 2 of you as a bench partner, 1 is a nurse, another 1 is a patient. We will do a throat culture, which means ask you open your mouth, say ah, then get a culture from your tonsil. then we streak onto the blood agar. Then we use a candle jar to do the incubation. Under micro aerobic environments, we put the candle there, then we light it, then we cover the jar, then it will be off. The light will be off, so which means most of the oxygen will be gone. That's micro aerophilic environment. Then after that, we find a pinpoint colony, most of them beta hemolytic. Then we do gram stem, we do latex agglutination to find out what's the bacteria. It's could be streptococcus, could be staphylococcus, which is on your tonsil area. That's what's the normal time we do. But we have to skip this semester because we cannot let you guys work too closely. So we keep another clinical lab, which is called the urine test. So that simply we can do, okay? So I just tell you, so we skip one clinical lab, but we can do a urine test. That's I usually say clinical lab 2. So first of all, when we talk about the urine test, we will talk about urinary tract infection. Acronym is UTI. Okay, what kind of the symptoms cause UTI? And very simple, that's called dysuria. Dysuria. Very easy, it is difficult to pee. So which means you may have a burning feeling during the pee, you may feel it's never end, or you have a difficult to take it out. Whatever, it's difficult to pee. Some of you have abdominal pain a little bit. That's called dysuria, which caused by called urinary tract infection. Okay, now why it will cause urinary tract infection? There is several reasons for that. We usually mention the reason cause urinary tract infection, including the number one is say being female, because the female, the urinary tract is short. So urinary tract is short. It's easy to get contaminated with bacteria. So female has a higher urinary tract infection rate compared to the male. The second one is holding on pee too long. So let's say you have a game between, it's not going to be this year, between WVU and the Pittsburgh, University of Pittsburgh for basketball game. You've been there as a studio for like 3 hours. You don't want to go to the pee, even you have a feeling about that. That is not good. If you keep doing that, the bacteria